This video is GPCRS1, G protein activation and deactivation. So here we're looking at the activation of a G protein through a G protein coupled receptor. And so the G protein coupled receptor, you can see up here at the top in green, it's a 7-pass transmembrane receptor, and a ligand can bind to it. This figure shows the ligand binding here. A lot of times I'm going to draw it binding here. This is presumably accurate for where it binds in terms of our biochemical understanding. However, just for simplicity's sake, I draw it up here. And so basically, while we have the 7-pass transmembrane receptor in its inactive state, then we have an inactive G protein. And you can see that there's 3 different parts of the G protein. It's a heterotrimer. And so there's this alpha subunit, which is shown in gold. This is linked to the membrane due to a lipid-linked tail. There's also a beta subunit, which is this kind of, I don't know, really light brown color. And that does not have a lipid-linked tail. However, the beta subunit is associated with the gamma subunit, which also has a lipid-linked tail. And therefore, the beta subunit is associated with the membrane. So to draw the abbreviated pathway, here we have the plasma membrane, the 7-pass transmembrane receptor, GPCR, and then we have the inactive heterotrimeric G protein. Which has the alpha, beta, and gamma subunits. And I would draw them in the same order and location, because then we can make our lipid tail here so that alpha and gamma have those links, whereas the beta subunit is not linked to the membrane. And finally, the alpha subunit in the inactive form is bound to GDP. Now I'm going to draw in red what happens when we have an activation of the signaling pathway. That's due to the ligand binding extracellularly. And then this recruits the heterotrimeric G protein to the receptor. This is due to a conformational change in the receptor, which then allows the heterotrimeric G protein to have a higher affinity for it. So to kind of write this out step by step, the ligand binds extracellularly. Triggering a conformational change in the receptor. The G protein is inactive and bound to GDP, can now interact with the receptor. And then GDP is exchanged for GTP. Which activates the alpha subunit, as well as beta and gamma. Okay, so let's write that out. So the third step, GDP is exchanged for GTP. So it's not just plopping off an inorganic phosphate, it's actually exchanging GDP and GTP. And this is due to the G-alpha protein's intrinsic ability to make that change. There's not another enzyme that triggers it, it's just G-alpha. At this time, then G beta and gamma will disassociate from G alpha, and they are also active when they're not bound to G alpha. And so it looks like they're somehow cleaving or like having a covalent bond broken between them. But remember that these are, each of these subunits is a separate polypeptide chain. It just means that they are disassociating from each other. So their non-covalent interactions are not interacting anymore. This step of the heterotrimeric protein breaking into the two different parts is shown down here. Here we see the active G alpha with GTP, and then we see G beta and gamma, which don't have anything added to them. It's simply that when they're released from G alpha, they are now active. And each of them can go off to activate their own effectors at this point. Which we'll hear more about later. And then just kind of reset this whole process and bring us back to the beginning. Again, this is not these G protein subunits are not permanently active. Instead, eventually GTP is hydrolyzed from G alpha. This means that G alpha is then in its inactive state, and then it has high affinity for G beta gamma, and so it can basically bind to them again and inactivate them. This figure 16-20, shows more of the inactivation or deactivation process. So here we're picking up with G alpha that's bound to GTP, and G beta and gamma, which are active. And basically they do their own thing, and they might be activating different effector proteins. Here we see this kind of baseball bat looking enzyme, which in future videos we'll see is adenylyl cyclase. Could be something else as well. So basically by GTP associating with whatever that effector protein is, then it activates it, which you see here. Eventually the inactivation takes place, and so that's this part of the figure. And so again, that's when one little inorganic phosphate is removed from the G alpha. This inactivates the G alpha subunit, which we see here. So that removal of the inorganic phosphate means that this is GDP, and now it's not exchanging GTP for GDP. It's removing an inorganic phosphate, which then produces GDP. And then now G alpha is inactive, and when it bumps into G beta and gamma, then it will form a stable configuration with them and thereby inactivate them.